All right, fine. You'll got the reading material. So today we'll cover up excipients and we'll also cover up this thing, the processing. Okay. Next lecture, we will understand tonicity. We will understand the important tests which are used for parenterals, especially BET or LAL test. And if time permits, numericals, वरना numericals का दूसरा class लेंगे. Okay, so this is our third lecture, right?
Yes, ma'am.
All right. Fine. So who's the admin today? At Pharmacad?
Ma'am, no man.
No man. Okay, so you will look at the waiting room, right? Keep on admitting everyone because I think the students are very less today.
Yeah, ma'am.
So just admit them. Okay, let me share the screen. Just a minute. Yeah, sorry to keep you all waiting. You all had some industry visit today to ACG. Hello. Chaitanya? All right. Anyways, fine. Then let's start. Let me know if you all can see the screen. Can you all?
Yes, ma'am.
All right. Today we'll have a relatively shorter lecture where we'll just do this part of because there are not many students right today. So we'll complete containers and closures, and yeah, I think we can do that much. And in the next lecture, we can do the processing and tonicity and all. All right.
Yes, ma'am.
So are you all coming from this thing, industrial visit right now? हाँ या ना, कुछ तो बोलो. Okay. Anyways, fine. Let's see how much can. Yes.
Actually, those students offline mode, they have gone to internship, and myself from the online mode right now. So we not able to go for the
Okay, you all couldn't go. So this is the online one, right?
yes, yes, ma'am.
Okay, 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 okay. Got it, got it. Fine. Anyways, we'll continue with our thing. Just a minute. So are you all understanding the topic?
Yes, ma'am.
Okay, good. Fine. Let's carry on then. So yeah, in today's class, we'll be studying about the containers and closures, which are an important component, right? In parenterals, you consider the containers and closures as a direct part of your excipient, not excipients, I would say, but your component. Okay. Under components, you will have your drug, you will have your excipients, but ye bhi bahut important aayega because again, your formulation is mostly in solution form, if not a lyophilized powder or a dry powder. And because of that, it's in direct contact. Okay. So when I'm talking about containers, I'm talking about the primary packaging. So you all understand what a primary packaging is, right?
Yes, ma'am.
What is a primary packaging?
Which is in directly contact with the drug itself.
Okay, that is a
directly contact with
okay. Yeah, fine. So primary packaging is the one which is in direct contact with your formulation, with your drug product, I will say. Understand DS आगे जाके तुम लोग को काम आएगा. DS मतलब ड्रग सब्सटेंस. ओके. ड्रग सब्सटेंस मतलब API. And DP is drug product. DP is drug product. माने के your formulation. Okay. So yeah, primary container is in direct contact with your DP. What about secondary container or secondary packaging? These are the different types of packaging we are talking about. Secondary packaging क्या होता है? Hello.
Ma'am, to pack those containers into box.
Yeah, the boxes. Okay. Now say your glass vial is your primary container and the box in which it is kept. Okay. The box or the carton in which it is kept, that is your secondary packaging. All right. Then your tertiary packaging. So primary packaging is kept in secondary packaging. Secondary packaging is kept in your tertiary packaging. माने के the bigger box in which you have all these smaller boxes kept. Okay. So this is your tertiary packaging. And probably you might have a fourth level of packaging, which is the big, that putha wala box aata hai na, that brown color cardboard carton. That becomes your fourth level. But primarily, we give, like, during studies, we study in this three packaging. Also, when you're doing stability study of your drug product, okay, drug product matlab formulation, you will do uska stability study in primary packaging as well as secondary packaging. Okay. Secondary packaging mein, especially this becomes important for photolytic studies and all of that, photolysis studies and all of that. All right. And ये जो सारे containers भी है ना तुम्हारे जो glass vials, okay, excuse me for my drawing, ये जो glass vials है उनको तुम सीधा भी रखोगे during stability studies and कुछ को ना तुम उल्टा करके भी रखोगे. So this is like the regular one, and this is one, the inverted glass bottle study. So can anyone guess ये inverted study? So here is your solution, and इसमें your solution will be here. So can anyone tell me सीधे में तो क्यों करते हैं normally drug stability and all that check करने में, but why is this inverted study also important?
Leak test.
Leak test, okay, in a way, okay, is it leaking from this thing? Okay, fine. Other thing?
The drug product is in contact with the closure. When it's
Very
involved.
good. Very good. To check if the not to check the drug product, now ulta करके रखेंगे तो it will come in contact with the rubber stopper, no? Okay. So तुम्हारे ग्लास वायल होता है, उसके ऊपर रबर स्टॉपर होता है, एंड उसके सराउंडिंग अल्यूमिनियम का सील होता है. तो अल्यूमिनियम सील से तो कांटेक्ट में नहीं होगा, बट हाँ जब तुम उल्टा करोगे तो ग्लास स्टॉपर के साथ तो इंटरेक्ट करेगा ना, राइट? So to check its interaction with the stopper system, or you can say the closure system. इसीलिए ये inverted test is also very important. समझा सबको?
Yes, ma'am.
Yes, ma'am.
Okay, good. These are some additional points which I'm telling you all, huh? And your stability conditions पता है ना तुम लोग को? One is at 2 to 8 degrees Celsius, one is at 25 degrees Celsius, 25 degrees Celsius, 40% RH. Third is 40 degrees Celsius, 75% RH. This is 40 or 60, I'm confused. एक बार you all can check. Okay, so these are the three main studies which are done. 2 to 8 को तुम 5 degrees भी बोल सकते हो. Fine. So yeah, let us look at the important characteristics and attributes of the containers and closures as well. So the container or the primary packaging should ideally be transparent to allow the product to be examined prior to use. It's important that if it's not photolytic, तुम उसको transparent रखो ताकि quality check में तो तुम particulate matter and all that you will check, but even the user before administering it, the nurse, or even if it's being administered at home, you can see, you can check अगर transparent होगा तो. Okay, so that is what will allow the user to do if it's a transparent thing. Fine. Ambered color में ये देखना मुश्किल हो जाता है, obviously, right? Ambered color, it becomes difficult. So this is about it being transparent. Having said this, if your drug is photolytic, then you will use what kind of vials? Huh? अगर तुम्हारा ड्रग फोटोलाइटिक है तो तुम कौन सा वाला यूज करोगे?
Amber color.
Ambered color. Very good. But you know there is one just a minute, I'll check the exact name. But there is one exception, you know. हमें ये तो पता है कि अगर it is photolytic, then it will be you will have to store in amber color. But there is one interesting example of this drug called as let me put it here, Pemetrexed. Okay, Pemetrexed injection. This comes as a liquid solution. Okay, it's a photolytic drug. Okay, it is a photolytic drug, but it comes in a transparent vial. Isn't that strange? It's a photolytic drug. It undergoes photolysis, but it comes in a transparent vial. So this is one interesting case study. Like I got to know by my professor lately, so I feel like sharing it with you all. So why isme, you know, why is it coming in transparent? It is because कि इसका precipitation का chances ज्यादा होता है if it is not stored in proper temperature conditions. Okay, fine. वो टाइप का उसका degradation का possibility ज्यादा है. Precipitation is also a form of degradation, yes or no?
Yes, ma'am.
Yeah. So वहां पे उसका possibility ज्यादा है. Okay. So अगर and photolysis भी होता है but वो कम होता है. And obviously you have your secondary container तो है ही. Correct? Secondary container तो है ही. So in that case you write the warning कि after you store it in the secondary container only. In case if it is a multi-dose container. Okay, to protect it from photolysis. But अगर ये amber color में आता तो amber color में precipitation देखने को easy पड़ता या would it be like easily visible if there's any precipitation?
No, ma'am.
No, right? So that is why the makers took a call, the innovators took a call to instead package it in a transparent one and compulsorily keep it in a secondary container. Got it? So ये जो हमारा थंब रूल होता है ना, फोटोलाइटिक ड्रग तो एम्बर्ड कलर में डालो. But when you go to industry and when you start working in R&D and all, there are multiple challenges which come. So there's one thing which is always said, कि formulation में there are no strict, no hard and fast rules. You make your own rules with proper justifications. Understood this example?
Yes, ma'am.
Yes,
Good.
ma'am.
Yes, ma'am.
Good. So this is the first thing. Then container must be tightly sealed to maintain sterility and prevent contamination, also leakage. Okay, so this is obvious. Impermeability. Your containers must be impermeable. बाहर से भी कुछ अंदर नहीं जाना चाहिए, अंदर से भी कुछ बाहर नहीं जाना चाहिए. Okay, they must be impermeable. And वो खुद भी कुछ रिलीज नहीं करना चाहिए. It shouldn't leach its substances into the formulation, वरना उससे भी contamination होगा. If it is not impermeable, वापस same problem होगा, sterility का, leakage का, contamination का, degradation through oxidation. Say if it is permeable to the atmospheric air, then oxygen can seep in and it can initiate oxidation. All right. Container must be inert. मतलब वो खुद प्रोडक्ट के साथ रिएक्ट नहीं करना चाहिए. It shouldn't react with the product. And again, it shouldn't leach out any substances into the product. All right. It should be robust to withstand the chosen sterilization process. Obviously, what do you think? Which process are they talking about? Which type of sterilization are they talking about here?
Autoclave.
Correct. Moist heat autoclave, or you can say moist heat sterilization. Suppose अगर dry heat होता रहेगा तो वो. Okay. Even radiation. हाँ.
Filtration करें.
What? Couldn't hear you properly. Even radiation, right? कि उस all these things it should withstand. अगर container ही उसमें ढीला पड़ गया, okay, then there will be a problem again, right? And then, yes, it should have uniform physical dimensions. This is with respect to the filling process, the ease in filling and packaging process. Because filling में you have a line, right? पूरा एक conveyor belt system होता है, पूरा line होता है. So it should be, I hope you all can see the things in red ink, right?
Yes, ma'am.
हाँ, filling के वक्त it becomes important. All right, uniform dimensions वो सब एक conveyor belt पे हो. See, industry will always prefer कि तुम same line में, same machines में 10 product बनाओ. ऐसे नहीं कि एक product के लिए मुझे एक set of instruments चाहिए, दूसरे product के लिए दूसरा set of instruments. That will become costly. Okay, so that is why these dimensions become very important for a simple process of filling also. कि एक ही filling line में तुम हर एक product का तुम fill कर सको. Got it? So then the packaging material mein, I told you there are two important components again. The first one is a container and the second one is a closure. All right. Containers are made of two materials or MOCs. Okay. The term we use is MOC. Mane material of construction. All right. MOCs. Then one you have glass and one you have plastic. The preferred one is glass because मैंने जो previously सारी चीजें बोली, more inert, it should be robust. Plastic I don't think is very much suitable for autoclaving. All right. And plastic में थोड़ा permeability भी ज्यादा होता है compared to glass. Okay. So glass is more preferred. Closures में you have your rubber stoppers or the plungers or the seals. And aluminium seals जो उसके ऊपर बैठता है, fine. So you have different types of containers. Let us first talk about containers. You have different sort of containers. We have ampules, right? So ampules पूरा एक seal जैसा होता है. You all can see this animation here. पूरा एक seal जैसा होता है. And then you break open from here. Okay, you break open from here. And then इसमें जो भी contents है, that you take it out via injection. So these are the ampules. And GNP matlab glass and plastic. ठीक है? So it's available as a glass. It's available in glass also. It's available as a plastic also. You have ambered color ampules also. Transparent भी होते हैं, tinted भी होते हैं. Tinted matlab they are not completely ambered. They are like greenish, yellowish tinge. Okay? That is a thing. So you have single-use ampules. Ampules are single-use. Okay, ampules are single-use because एक बार break करने के बाद we cannot seal it back, right? Can you all hear me? Is there some lag or anything?
No, ma'am.
Okay. Ampules are single-use only and 1 to 10 ml के range का आता है ampules. All right. अभी उससे बड़ा में अगर जाना है, if you want to go a little more in that, then you go for vials. All right. And vials are usually glass only. Vials are glass only. I have not seen plastic vials. Ampules you get. You have 1 और 2 ml का भी कंटेंट तुम भर सकते हो. 2 ml के भी vials आते हैं. All right. But your book says 5 to 100 ml. Let us go with that. But 2 ml के भी vials आते हैं. Then you have syringes. Okay. So this is one process. Okay, yeah. Let me tell you all. This is a blow and fill seal process of making of plastic ampules. So plastic ampules में कैसे है? तुम ये extrusion mold में you blow in your polymer. Okay. You blow in your plastic ampule. उसको inflate करेंगे and then उसी में भरेंगे तुम्हारा content and then वहीं पे seal करेंगे and then you get your finished product. Okay, so एक ही line में you will blow in the inflate your this thing ampule, fill the content, seal it, and ये पूरा under sterile aseptic conditions होना चाहिए. All right, this is a process for plastic ampules. Yeah. Then you have your syringes. Syringes you all know, right? Injection, जो हम बोलते हैं. That is your syringe, and then you have needle. And needle का हमने different gauge size and वो सब as an assignment पढ़ लिया है. तुम लोग के सारे assignments check कर लिए, right? Online students.
Yes, ma'am.
You got it. We'll discuss at the end of the lecture the assignments.
Okay.
उसमें doubts है किसी को कुछ? होना चाहिए doubt उसमें. Anyways, then we have cartridges. अभी cartridges क्या होता है? It's a new system which is called as the pre-filled syringes. All right, pre-filled syringes, as the name suggests. Normal syringe कैसे? तुम्हें ampule या vial में से तुम्हारा formulation लेना है and then you will inject it. Pre-filled syringes मतलब syringes जिसमें तुम्हारा product is already filled. Okay, so those are pre-filled syringes. And that comes with a plunger is just say, you push it through, and then a needle. So there are 3 components in a syringe. All right. Yeah, there are 3 components in a syringe. First, you have your main syringe body. Okay, then you have a plunger. Okay, this is the syringe. And then you have a needle. Okay, so there are 3 systems, 3 components to your syringe. So, abhi pre-filled syringe mein matlab this part is already pre-filled. You all can see this, right? And this is called as a cartridge. So, in a pre-filled syringe, jo ye part hota hai, usko tum cartridge bologe. Understood? So, this is a cartridge. Iske पीछे हम plunger लगाएंगे. Okay? And idhar hum needle लगाएंगे. This is the thing. Now there are certain pre-filled syringes with dual compartment. Okay, so ये एक cartridge में तुम्हारे 2 compartments होंगे. Okay, एक rubber septum से separate किया होता है. Okay, rubber septum से separate किया होता है. तो इसमें क्या होता है, तुम एक बार okay, पहले तो अलग से plunger attach करोगे. एक बार push करोगे ना, तो ये दोनों कंपार्टमेंट 1 and 2 का कंटेंट्स मिक्स हो जाएगा. सो द जो भी प्रोफेशनल जो एडमिनिस्टर करने वाला है, एक पुश करेगा, इट विल ब्रेक ओपन, मिक्स करोगे, एंड देन यू पुट योर नीडल एंड देन इंजेक्ट इट. ऑल दीज नाटक ये जो करना पड़ता है, दैट इज बिकॉज ऑफ ease. Okay, imagine mixing 2 ampules and then giving it, so it becomes easy. Most of the issues are because of compatibility and stability. Already अगर pre-mix करके ही हम देते, it would be much more easier, but then there might be some issues of compatibility and stability. That is why उसको separate करना पड़ता. Okay, and because it's again a low dose. Okay, अगर दोनों ड्रग को अलग अलग मिक्स करके देना है, तुम आईवी बैग से भी दे सकते हो. आईवी बैग में पहले वो वाला डाल दो, देन यू पुट योर सेकंड फॉर्मुलेशन. बट व्हाट इफ यू जस्ट वांट टू गिव 10 ml टू द पेशेंट? In that case, all these things you have to do. Are you all understanding? So all of this becomes important with respect to the stability, compatibility, and basically the requirement. And then comes the ease at the end. Okay, and if you start working marketing your product based on ease, that is when you develop new technologies and make more money, monopoly in the market. समझ आ रहा है सबको? Are you all keeping up?
Yes, ma'am.
Yes,
Like,
ma'am.
I hope like you all don't find it कि अरे इतना ज्यादा extra हो रहा है ये वो and all. If it's so, let me know. I'm trying to give all what I've gathered. Is it like that?
No, ma'am.
Okay. I hope you all are enjoying the lecture then, in that case. Okay, fine. Then comes the bottles. Okay, which these are basically, now these 2 things are basically your which type of formulations? Look at the volume.
Large volume parenterals.
Perfect. These are your LVPs. All right. So इसमें preservative होगा नहीं होगा?
नहीं होगा.
Good. So you do not have preservative in here. All right. So these infusion bags तो plastic के ही होते हैं, collapsible. I recently came across a few patents wherein they have dual chamber infusion bags also. Okay, dual chamber infusion bags. उसमें दो separate चीजें हैं. You open something or you plunge it something and then the contents get mixed. So you have वैसे चीजें also. All right. Then bottles, glass and plastic के होते हैं. And this is for like 100 ml to 1 to 3 liters का होता है. And these are the two polymers being used, PVCs and polyolefin plastics. All right. So this was about containers. Yeah, this is basically what I was trying to tell you. The needle, the barrel. Okay. This is the barrel and this is a plunger. ये भी PFS है, pre-filled syringes. So pre-filled syringes में ये सब attached आता है. Cartridges में तुम्हें attach करना पड़ता है. Okay, they come separate. Yeah, so this is a little theoretical part here. Okay. Yeah. So glass is mostly the material of choice for your small volume parenterals. Okay, large volume में वो convenient नहीं होता है. And your glass is composed of, this is an important point, please make a note of this point. It is composed of your silicon dioxide with varying amount of other oxides. So this is your primary component of glass. And then you have other oxides of sodium, potassium, calcium, magnesium, aluminum. Boron is another important one here. Okay. And iron. All right. So these are the components of your glass. So at times what happens if your glass is not of good quality, if it's not manufactured properly, and say you have your solution. Okay. You have your solution in it. Over the time, all these oxides will leach into. बाहर से कुछ नहीं आ रहा है. ये जो अंदर ही तुम्हारा ग्लास के अंदर ही जो सब है, that all is going to come out. Come out as in it's going to come out into the formulation. Okay. External environment से कुछ नहीं आ रहा है. ये इसमें से ही आ रहा है. So this process is called as leaching. And this is definitely not favorable. You don't want this to happen. But ये होता है कभी कभार. So तुम whenever you're selecting a glass and all of that, compatibility में leaching studies and ये सब भी तुम test करते हो. ठीक है? So yeah, this is the thing. Then you have the basic structural network of glass is formed by the silicon oxide tetrahedron. This is a potential MCQ. Okay. Basic structural unit of glass. It's a tetrahedron formed of silicon oxide. All right. And these migratory oxides, which is called as the leachables. Okay. Migratory because they can migrate into your formulation, may be leached into a solution in contact with the glass, particularly during increased reactivity of thermal sterilization. Okay, so this migration or the process of leaching is enhanced by the process of heating. All right. Not only heating, even pH conditions. Even certain pH conditions, अगर pH conditions formulation में वैसे है कि it is good, it is favorable for these oxides to get into, then that also happens. All right. So that is what happens. Now the oxides dissolve may hydrolyze to okay, so formulation के अंदर का pH है जिससे भी ये सब promote हो रहा है leaching, and once these oxides get leached into your formulation, it is again going to alter the pH of your solution. Again तुम्हारा solution का pH अगर change हुआ, so जो भी तुम्हारे drug का pH dependent solubility है या फिर हाँ ड्रग का पीएच डिपेंडेंट सॉल्युबिलिटी है या फिर जो भी स्टेबिलिटी है ये वो एंड ऑल ऑफ दैट इज गोइंग टू बी प्रोन टू डिग्रेडेशन, राइट? इट्स गोइंग टू enhance or catalyze the degradation, right? So leachables are that is why leachables aren't good and it can lead to degradation of your products. Okay, and some glass compounds will be attacked by solution and in time dislodge the glass flakes. Okay, okay, so ये flaking भी हो सकता है, मतलब तुम्हारा ग्लास ही एज फ्लेक्स निकल रहा है, बहुत पतला पतला, okay, into the solution, and such occurrences can be minimized by the proper selection of glass composition. कि तुम कौन सा टाइप का ग्लास यूज कर रहे हो, ओके, कोई एडिशनल कोटिंग चाहिए तुम्हें, is your formulation compatible? So all of this becomes very important. हम tablets and capsules में compatibility study नहीं करते with the packaging, but here you see how important the packaging is. All right, so did you all understand the point?
Yes, ma'am.
Hello. Yeah, this brings us to the classification of different types of glass as per the USP. So you have type 1, type 2, type 3 and type 4. Okay, type 4 is non-parenteral. That is why like I've written NP here. It's not parenteral. Okay, so the first 3 types are the major ones. Let us look into it one by one. So type 1 is a borosilicate glass. It's used for injectables. It's the best amongst all. Okay, it's highly resistant to your autoclaving temperatures. It is highly resistant to leaching and all of that. Okay, so type 1 are the most preferred glass. All right. So this is the thing. I told you, right? Boron oxide is very important. So you have 81% of your silicon dioxide, which is the main tetrahedron structure. Then you have boric oxide, and then you have aluminium oxide. And with it, you might have some little composition of sodium, potassium, magnesium, and all as well. All right. Then you have maximum chemical resistance, as I told you. Chemical resistance, matlab it's highly compatible to a wide range of pH, low leachability, low thermal coefficient of expansion. माने के it won't expand under heating condition. In simpler terms, it is stable at higher temperatures. Heating का इस पे ज्यादा effect नहीं होगा. All right. Then is type 2, which is called as the soda lime treated glass. It's important to remember the names. So type 2 is your soda lime treated glass. So it has relatively high proportions of sodium and calcium oxide. सिलिकॉन डाइऑक्साइड तो होता ही है, बोरिक ऑक्साइड भी होगा ही, बट आई टोल्ड यू ना देयर आर in type 1 there are some amounts of sodium and all of this. So here the proportion of these lesser quantities is a little more. All right, so you have more amounts of sodium and calcium oxide. That is why इसके leachability की possibility भी ज्यादा है in type 2. All right, so they are chemically less resistant. And type 3 is your normal soda lime glass. This is your soda lime glass and this is your soda lime treated glass. We'll understand what's the difference. So this type 3 is usually used for oily preparations. Mane oily preparations matlab non-aqueous preparations. So zyada reactivity, because type 3 is सबसे less stable, हम उसमें ऐसा ही कंटेंट भरेंगे जो जिसकी रिएक्टिविटी कम होगी. नॉन एक्वस चीजों की रिएक्टिविटी इज यूजुअली लेसर कंपेयर टू एक्वस, कंपेयर टू वाटर बेस्ड. ऑलराइट, सो इसमें ऑयली बेस्ड प्रिपरेशंस तुम फिल कर सकते हो एंड देन रिकॉन्स्टिट्यूटेड प्रिपरेशंस फिल कर सकते हो. माने कि ड्राई पाउडर के ड्राई पाउडर बस फिल कर दिया, ओनली एट द टाइम ऑफ एडमिनिस्ट्रेशन तुम उसको रिकॉन्स्टिट्यूट करोगे. वो लायो पाउडर को, लायोफिलाइज पाउडर को, at the time you will reconstitute and immediately you will administer. So the amount of time, the residence time of your formulation in this container is going to be minimal, correct? So that is why type 3 में तुम ये दो type के products fill कर सकते हो. All right. So they melt at lower temperature and they have a higher thermal coefficient of expansion. माने कि they are less stable at higher temperatures. All right. So now let us understand what is the difference between your soda lime glass and soda lime treated glass. So, yeah. Soda lime glass is basically something made of your soda lime. All right. Now, when you coat the inner surface, just a minute. Yeah. So when you coat the inner surface of your type 3 glass with what? With moist SO2, with moist sulfur dioxide at a temperature more than 500 degrees Celsius. Okay. It neutralizes the surface alkali. Soda lime, you all understand, right? It's alkali only. Your NaHCO3, correct? If I'm not wrong. Soda lime. So that is the thing. It is alkaline in. Hello. Yeah. Huh? Is anyone saying something? Okay. Anyways. So soda lime is alkaline in nature. All right. You want your glass to be neutral. So wo alkaline nature wale surface ko you will treat with moist sulfur dioxide at a temperature more than 500 degrees Celsius. So ye neutralize ho jayega. Thus the surface in contact with formulation now is neutral. Hence this type of glass is suitable for injectables. However, repeated use may not be possible. Okay. So type 2 is not used in LVP and also for multi-dose. Also for multi-dose vials. MDVs में भी multi-dose के लिए भी उसको नहीं prefer करेंगे. One time use के लिए prefer करेंगे जैसे कि ampules के लिए. And obviously I hope you all understand कि type 3 सबसे सस्ता है, type 1 सबसे ज्यादा महंगा. All right. So this is one important thing which you all need to know. The difference between type 3 and type 2 and how is type 2 formed. Soda lime treated का मतलब क्या हुआ? All right. And type 4, as I told you all, it is soda lime glass only, but not suitable for containers for parenteral use. All right. So those were the things about glasses. Did you all understand that? Different types of glasses?
Yes, sir.
Yes, ma'am.
Okay. Then accordingly, कौन सा glass है वो भी तुम्हें test करना becomes important, right? So accordingly, you have different tests, and these all are given by USP. Even this classification was based on US Pharmacopoeia. Even this is based on USP. Okay. So you have two major tests. You have a powdered glass test, and even this is important, all right? You have powdered glass test and you have water attack test. So powdered glass test, as the name suggests, तुम glass लोगे, उसको powder कर दोगे, and then you will test its क्या क्या लीचेबल्स है. तुम बेसिकली टाइट्रेशन करके फाइंड आउट करोगे कौन सा ऑक्साइड का कंपोजिशन proportion कितना है. Basically you will find out the composition and all of that. All right. That is done for type 1 and type 3. Okay. That is leaching potential of the interior structure of the glass तुम्हें पता चलेगा. What you get to know. The objective of powdered glass test is this: to understand the leaching potential of the interior surface of the structured glass. And this is the method on the USP on. USP के general chapters होते हैं ना, उसमें से मैंने लिया है. Then you have water attack test. Now this is done for the intact surface of the container. Abhi, सपोज अगर हम टाइप 2 ग्लास लिया विच इज द ट्रीटेड ग्लास, उसको इफ वी क्रश इट देन वो सारा कोटिंग भी निकल जाएगा, देन तुम्हें जो टेस्टिंग का रिजल्ट आएगा दैट वोंट बी ट्रू, राइट? सो यू डोंट डू द पाउडर वाला टेस्ट, पाउडर्ड ग्लास टेस्ट फॉर टाइप 2. ये इंपॉर्टेंट है ध्यान रखने को कि पाउडर्ड ग्लास टेस्ट इज नॉट डन फॉर टाइप 2 बिकॉज उसमें तुम्हारा कोटिंग डिसरप्ट हो जाएगा. All right, and you won't get true results. You won't get true results. So type 2 के लिए you do water attack test. So what you do is you take the container, type 2 container, you fill in your formulation, test, and then you test the formulation. कि formulation में कुछ leach हुआ है क्या? Another way, okay, this is one way. Not one way, this is one step. The second thing is where you again take your container and you fill in water in it. Plain water you fill. इसमें तुमने DP भरा था, यहाँ पे तुम water भर रहे हो. I hope you all understand DP when I'm saying DP is your API containing formulation. यहाँ पे तुमने water भर दिया and then you test that water. बेसिकली उसका भी टाइट्रेशन करके यू फाइंड आउट के कितना कंटेंट लीच हुआ है एंड ऑल. सो दीज आर द टू इंपॉर्टेंट टेस्ट. इज इट क्लियर एवरीवन?
Yes, ma'am.
एंड व्हाई वी डोंट डू पाउडर्ड ग्लास टेस्ट ऑन टाइप 2 वो भी क्लियर है, करेक्ट?
Yes, ma'am.
Yeah. Now we looked at the advantages of glass, right? Let us look at the loopholes. The first major one is leachables and extractables, जो मैं तुम्हें बोल रही हूँ. Because your glass is made of these oxides and dioxides which can leach into your formulation depending on the pH of your formulation and all of that. वो leaching का possibility increases, all right? So let us look at what are leachables and extractables. It's very important to understand the difference between the two. Now imagine this entire thing, okay, the entire circle is your container. Okay, entire is your container. So तुम्हारे container में extractables भी होंगे, leachables भी होंगे. But when you fill the formulation, okay, this is your container again. But जब तुम्हारे surrounding में जब तुम्हारा formulation होता है, then the one which gets into, which migrates into your formulation, that is called as leachables. So leachables have the ability to migrate. Okay, they can migrate into your drug product. Extractables don't migrate. All right, they are there. In order to test extractable, you need to extract them forcefully. That is why they are called extractables. Leachables, they automatically leach in your formulation. All right. So extractable studies are an assessment performed on the material. All right. And leachables are the study which is performed on your DP. To study your leachables, तुम container में तुम्हारा DP भरोगे. You will keep it for some time. You will expose it to heat or different pH or whatever. And then you will study तुम्हारा, इसका तुम ऐसे करोगे, इसका तुम टाइट्रेशन करोगे तुम्हारे डीपी का, एंड देन यू विल सी कितना लीचेबल्स है. बट एक्सट्रैक्टेबल्स तो मूव नहीं हो रहा है, एक्सट्रैक्टेबल्स तो माइग्रेट नहीं हो रहा है, वो वहीं का वही है. बट इफ यू वांट टू स्टडी योर एक्सट्रैक्टेबल्स कौन से है, देन यू डू अ टेस्ट ऑन योर पैकेजिंग मटेरियल, ऑन योर कंटेनर. इज इट अंडरस्टूड एवरीवन? वेरी क्लियर?
Yes, ma'am.
Doubts anybody? अभी के अभी पूछ लो. Leachables can migrate. They migrate into your DP. That is why to analyze it, you need to study DP. Extractables don't migrate. They stay in your container. That is why to study it, you need to test your container. And it is applicable not only to glass, even to plastics, even to your PVCs, but the possibility is more in case of glass. Clear, everyone?
Yes, ma'am.
Good. Yeah, so these are some pointers here. Okay, they have relatively low levels. There should be relatively low levels of leachables at pH 4 to 8. All right. High levels of leachables above pH 9 आता है glass में. Major extractables are silicon and sodium. Minor ones are these in a glass, even boron. Then trace major, minor and trace मतलब और भी ज्यादा कम में you have iron, magnesium, zinc. And treated glass vial. Treated glass vial मतलब कौन सा type?
Type
Type 2.
Yeah. They give lesser extractables if the pH is less than 8. These are just some pointers which you all need to remember. इसमें कुछ logic. ठीक है?
ma'am.
Yes, Chaitanya.
For extractables, is there any specific test that we do as we do for the leachables so that no leaching will takes place?
हाँ,
Like
leachables
that?
के लिए तो तुम तुम्हारा DP एनालाइज करोगे, करेक्ट? ड्रग
Yes.
प्रोडक्ट एनालाइज करोगे. आई डिडंट अंडरस्टैंड योर क्वेश्चन. एक्सट्रैक्टेबल्स के लिए तुम तुम्हारा पैकेजिंग मटेरियल का एसे वगैरह करो.
So means the main solution that is present that will analyze.
मेन सोल्यूशन नहीं. फॉर एग्जांपल तुम ग्लास का तुम एक्सट्रैक्टेबल टेस्ट करना है तुम्हें, तो अगेन वो पाउडर ग्लास टेस्ट करोगे.
Okay.
All right, that you will.
Yes.
Then is your delamination. Delamination is nothing but the flaking of the glass. All right, flaking of glass. That is your delamination. तुम्हारा glass will keep on coming out. There's one term which I'm missing. Yeah, it's that one. Okay, so that is your delamination. Then is adsorption. Adsorption is nothing but, मैंने कौन सा एग्जाम्पल दिया था लास्ट क्लास एड्सॉर्प्शन का? उसका असाइनमेंट भी पूछा था मैंने. डैश गेट्स एड्सॉर्ब्ड ऑन द ग्लास, दैट इज व्हाई यू कोट द ग्लास कंटेनर विद डैश. कम ऑन, नोबडी. प्रोटीन्स का बोला था ना मैंने.
Bovine serum albumin.
Yeah, proteins get adsorbed on the glass, and that is to prevent that. In order to prevent that, you coat it with BSA, bovine serum albumin.
Yeah.
You all forgot also.
No,
Adsorption मैंने तभी का तभी explain किया था. याद आया? And then another problem are your cracks and scratches. Your glass is prone to cracking, scratches, and all of that. But the major ones are these. First is this, then is this, and then is adsorption. All right. So these are some loopholes about your glass. Then comes the plastics. So plastics which you use are thermoplastic. मतलब वो heating पे उसका shape change हो जाता है. Okay, that is what is thermoplastic. They have been established as your packaging material for sterile preparations, especially for your LBPs. Then your ophthalmic solutions का भी bottle वैसे ही आता है. Increasingly, and now it's being used in SVPs, but believe me, by and large, it is LVPs only. All right. Permeation of vapors and other molecules. Now, these are some important pointers with respect to glass. Permeation of vapors and other molecules in either direction through the wall of plastic container. ये होता है. मैंने impermeability का भी point explain करते वक्त I told you कि प्लास्टिक में इसका chances ज्यादा है because the pore size, the molecular pore size in your PVCs and all is larger than glass. So that's why permeability का issue in case of plastic ज्यादा होता है. Second is leaching of the constituents. And that is why you overwrap it with a primary container. Then is leaching of the constituents from the, and you have coated plastics also. Plastic hair normal, and then you further coat it also, maybe on the inside also, outside also. Then is leaching of the constituents from the plastic into the product. Obviously, leaching, I told you, it happens both in glass also, here also. Understand? What is the difference between permeability, this point and this point? Leaching में and ये permeation में difference क्या है? दोनों में तुम्हारा product में आ रहा है. But what is that two differences? Anyone?
Outside permeation is out from the outside of the container, and leaching is from the packaging materials.
Very good. लीचिंग की अगर हम बात करते हैं तो लीचिंग इज कंटेनर का जो कंपोजिशन है उसके कंटेंट्स तुम्हारे प्रोडक्ट में आ रहे हैं. परमिएशन में एनवायरनमेंट से अंदर आ रहा है. एंड अनदर पॉइंट ऑफ डिफरेंस,
Also the leaching is of the basic oxides through which the leaching takes place.
हाँ मतलब you needn't be so specific, but this was the major thing. Permeation is from the environment, leaching is from the container. Another point is leaching is one way, from container to formulation. Permeation is two way, environment to product and product to environment also. All right, in form of leakage. So permeation is two way. Then sorption in form of absorption or adsorption of drug molecules or ions on the plastic material. Same point. So you select the material based on the compatibility studies. Very important. All right. And leaching को भी prevent करने के लिए you add few additives. Your chelating agents here come into play, huh? Chelating agents. Why is it going so slant? Chelating agents. Example?
EDTA.
EDTA.
EDTA. Okay. So this is some, so that will chelate with all these metal oxides and all. So same problems you have in plastic also. One different one is permeation. Again, advantages, disadvantages. Plastics do not break. They are lightweight, easy to carry. And that is why, and that it is collapsible. So that is why तुम्हारे LVPs में use होता है. Flexible है. All right. Disadvantages, it's opaque. It's not as clear as your glass vial. So in case of some visual observation or visual inspection, it becomes very difficult. And obviously because they are plastics, they are unfit for thermal sterilization. तुम्हें कौन सा स्टेरिलाइजेशन करना पड़ेगा? कोल्ड स्टेरिलाइजेशन. कोल्ड स्टेरिलाइजेशन को क्या बोलते हैं? फिल्ट्रेशन. और एल्स रेडिएशन. बट रेडिएशन तो सिर्फ तुम्हारा सरफेस ही करेगा. And then third is your aseptic processing. But aseptic processing is cost. All right. So basically, these are the challenges with your using plastic as your container. Now coming, so any doubts in plastics? Huh?
No, ma'am.
All right. Now coming to rubber closures. So these are the basic. All right. First, you'll look at the diagram here. तो ये तुम्हारा glass vial है. उसके ऊपर तुम तुम्हारा रबर क्लोजर लगाते हो एंड देन ये अल्यूमिनियम सील लगाते हो. दिस इज कॉल्ड एज अल्यूमिनियम सील. एंड इस पे अगर और एक ऐसे करके एक टॉप अप होता है एंड दे आर कलर्ड ओनली. ऐसे ही एग्जैक्टली दिखेगा. ओके. ये अल्यूमिनियम सील के ऊपर भी एक अलग अलग कलर के ऐसे आते हैं फ्लिप्स. So these are called as flip off seals. So if your aluminum seals come with a plastic flip off, which is colored, then it is called as a aluminum flip off seals, or it's called as a flip off seal. If it is just your aluminum seal, it's called as your aluminum seal. Okay. So the तुम्हारे क्लोजर सिस्टम्स में ये 3 चीजें आएगी, रबर स्टॉपर्स, अल्यूमिनियम सील्स एंड अल्यूमिनियम फ्लिप ऑफ सील्स। ऑलराइट, सो वो फ्लिप ऑफ को तुम पहले फ्लिप ऑफ लिटरली ऐसे पील करोगे, फ्लिप करोगे, देन अल्यूमिनियम सील को डिक्रिम्प करोगे एंड देन यू विल पुट द इंजेक्शन, और इस अल्यूमिनियम को भी वैसे ही रखोगे एंड देन जस्ट you will pierce the needle through the rubber stopper and go. So here you have different seals here. Mostly gray color के seals use होते हैं. White and red, red usually LVPs के लिए use होते हैं usually. White and all they are used for some special purposes. All right. Now this one you see, this one, this one. These are your normal seals. Okay, normal seals. ये and even this one. इसका कुछ स्ट्राइक हो रहा है, मैंने लास्ट क्लास एक्सप्लेन किया था. इसमें एक ग्रूव है, वो ग्रूव किस काम आएगा? हिंट, फॉर द एस्केप ऑफ वाटर वेपर. कौन से प्रोसेस में काम आएगा?
Lyophilization.
परफेक्ट, वेरी गुड. इन केस ऑफ लायोफिलाइजेशन मैंने बोला ना, हाफ सील करेंगे और वो सील में ग्रूव होगा. सो दिस इज द ग्रूव, सी. सो इधर से वो एस्केप होगा. सो एट टाइम्स यू हैव अ सिंगल ग्रूव or you have a multi groove like this. You have igloo stoppers, you have a dual thing. Okay, एक ऐसे होगा and एक ऐसे होगा. So इधर से निकलेगा and वो सामने वाली साइड से भी. So you know, these are the different designs of your lyo stoppers, lyophilization stoppers. So based on your use, you will use that. All right. किसी में ज्यादा water vapor तुम्हें निकालना है in कम time. So in that case you will use ये वाला, this one. इसमें 4 slits है जहां से ज्यादा निकलेगा. Okay, water vapor. So these are the different types of rubber closures. Now here you need to understand the ingredients. This you need to remember for MCQs. Elastomers में कौन से आते हैं? वल्कनाइजिंग एजेंट और क्योरिंग एजेंट. इसमें तुम्हारा मेन तो सल्फर ही होता है, पेरोक्साइड भी होता है, कम यूज होता है. मेन सल्फर इज योर वल्कनाइजिंग एजेंट. It helps in the cross linking of the polymers. Vulcanizing agent basically strengthens your polymer. It cross links, it bridges the polymers. Then you have the accelerator, it's used for the process to speed up the process. Then you have activators, antioxidants, all right, because all of these can undergo oxidation. You need to have some antioxidant. Plasticizer and lubricant you have for the plasticizer and lubricant, because you need the stoppers to be coming from filling line mein. For the easy movement of stoppers, you have a coating of paraffinic oil or silicone oil. That is why if your rubber closure is also not made properly, if your rubber stopper is not made properly, तुम्हारे product में silicone oil का contamination आ सकता है. So that is why उसको further भी coated Teflon से किया जाता है, rubber stoppers को. Okay, so this plasticizer and plasticizer as a contaminant is actually a common thing. Okay, so it's been taken care of. Then you have fillers. Fillers are nothing but like your diluents in your tablets. And pigments or inorganic oxides. Carbon black is used for the gray color. Iron oxide is used for the red color. Titanium will be used for white color. Okay, titanium dioxide, TiO2. All right. So this is the thing, and this is the process. You all can go through it. Yeah, these are some physical characters. This is one important point. मैंने जो वो बोला ना, you have a Teflon and Fluorotec coated rubber stoppers. These are the different coatings in a stopper. So यहाँ पे तुम्हारा leachability यहाँ पे भी आएगा. Obviously, you saw so many things are going into your rubber stoppers, and that is why वो inverted test हम करेंगे. Inverted vial test during stability to check the reactivity and compatibility of your DP with the stoppers. Elasticity होना चाहिए. By elasticity, hint. Elasticity is like if I'm pulling it and leaving it, so it will again come back to its original shape. Come on, use your brains. Why elasticity? Because हम rubber stopper को we are going to pierce it through with the syringe or with the needle. Okay, in case of multi-dose vials, हम ये rubber stopper को we are going to pierce it through the needle. And when we remove it again, it should again, because of its elasticity property, it should again close that pore. All right, it should again close that pore. Close नहीं हुआ तो क्या problem होगी?
Contamination might.
Contamination, sterility तुम्हारा problem हो जाएगा. So that is why elasticity is a very important thing in your rubber closures. And that is why वो flip off seal भी होता है in multi-dose. कि एक बार flip कर दिया, you can again seal it also. Okay, some of them are like that also. And minimal fragmentation. Again, link it to the thing. Minimal fragmentation का मतलब when you are piercing it with the needle, वो rubber stopper टूट के अंदर नहीं चला जाना चाहिए तुम्हारे प्रोडक्ट के. हो सकता है, but it should be very minimal. There should ideally be no fragmentation. Understood? Because it is like a defense membrane, right? So it should again close back once तुम्हारा needle pierce होके अंदर गया, बाहर आया, it should close back and वो piercing action के वजह से fragments नहीं बनने चाहिए. All right? Hardness should be appropriate and controlled vapor permeability that I've explained here. So is everything clear about rubber stoppers? Any doubts? Hello?
No, ma'am.
Okay, fine. Should we start the second topic? I think we can do that. कि पहले assignment discuss करना है.
I will discuss assignment.
Okay. We'll discuss assignment and if time permits, we'll initiate the processing part. ठीक है?
Okay.
You all can't see the screen, right?
No, ma'am.
Yeah, can you all see it now?
Yes, ma'am.
Fine. So question 3 was, and please one request, order में लिखो. 3, 4, 5, 6 है तो order में लिखो. तुम लोग कैसे भी लिखो, but while scanning, at least scan question 3 पहले, then 4 पहले, 5 पहले. It becomes very hectic for me to then, scroll through. Okay. First is the use of physical stabilizers in biologics. Some of you all, and this is in general, not only for the online batch, but also for Dadar and Thane branch. किसी ने कुछ भी लिखा है stabilizers में. I specifically asked biologics के लिए, and I was expecting the answer of the proteins one, which I had discussed,
Yes,
right? Proteins and the bovine serum albumin ka coating and all of that. So uske liye, those who've written that have given 1.5 1 mark and additional information ke liye bhi 1 mark. So for that. Then function of inert gases. Inert gases maybe you'll have to explain it is used as an antioxidant. Why is it used as an antioxidant? Because वो oxygen को displace करता है. Connect those things. Cryoprotectants used during lyophilization. Why? सबने बस प्रोटीन का लिखा है, but it's used for small molecules also ना. Freezing process को nano particles liposomes का मैंने explain किया था. Nobody even mentioned about it. Fine. उसका भी चाहिए था, सिर्फ proteins के लिए नहीं. It's used for small molecules also. An example, some of them have literally pasted it from the internet. DMSO लिखा है, ethylene glycol लिखा है. Those are not used. These are the ones used. Sucrose, trehalose. इसमें से किसी ने भी दो at least लिखे, मैंने I've given 1 mark. So I don't know. This assignment wasn't really good. And process of lyophilization, mention it's based on sublimation का principle. Triple point of water mention कर दो. 3 steps mention कर दो. इसमें से कोई भी 3 steps है, used to enhance stability. Hydrolysis, oxidation भी prevent होता है. There were so many points to discuss. Some of them have discussed it very nicely. Some of them have, God knows what have you all written. Okay. So this was about the assignment. Anyone, any doubt? Hello.
Ma'am,
Yeah, Chaitanya.
in case of inert gases question, what exactly we have to explain about the, for example, if nitrogen is used, then what was expected in the answer part?
पहले तो oxygen environment का oxygen होता है during filling. Okay. That can lead to oxidation, which can lead to degradation of the product. So that is why you purge it with N2. Inert gas like N2 or some other gases also. There are some people who give an example of other gases also. Okay, you purge it with, so वो headspace. These are the keywords. Headspace का oxygen displace होता है. अंदर से भी अगर कुछ oxygen होगा solution में, वो भी displace होके N2 inert environment create हो जाएगा, which मतलब oxygenless हो जाएगा and or minimal oxygen, and that will have an antioxidant effect. I made a mention about one instrument also, na, called as oxybaby.
Yes, yes.
Which is used. Nobody even mentioned about it. So when I give you all assignments, it's not that you all have to always open books or go through internet. Use like मैं जो lectures में जो explain करती हूँ, please go through those things also. All right?
Yes, ma'am.
Fine. So yeah, this was it. So I think we'll end up here and we will start the processing and tonicity in the next class.
Yes, ma'am.
All right. And if time permits, next class we will do numericals या फिर उसके बाद ही हम numericals कर लेंगे. ठीक है, एक अलग क्लास में लेकर.
Okay.
So everyone, any doubts as of now? And today, because it's just 1/3 of the batch, no assignment for you all today. All right. So enjoy, but see to it you all study this thing. Read it from Remington. Lackman se processes. I'll prefer Remington only. Fine. Any doubts? Krutika, Sheetal,
No,
Tahir,
No, ma'am.
Tahir, Reshma, Nikita. All right. Sejal.
No doubts.
Okay, fine. Are you all understanding everything? Are you all keeping up?
Yes.
Good.
Yes, ma'am.
Fine. So इसका टेस्ट होगा तभी देखेंगे कितना समझे हो तुम लोग. Fine. चलो then. Good night everyone. Bye bye.
Thank you, ma'am.
Thank you, ma'am. Bye.